well, how do we even treat Cryptococcus neoformans? Well, there are actually 5 main types of antifungal drugs for this pathogenic fungus, but intrinsically, it's actually already resistant to 2 out of those 5. So that only leaves like 3 of these classes left in terms of treatment, and that can be a problem if you're, like, for example, trying to combine treatments, or resistance could also develop in those other classes, and 3 classes isn't even that much to play around with. So with that, here we basically have a Cryptococcus on the left, a smiley face, and a happy human on the right. And if we want to try to find more of these actual antifungal drug treatments, it can be a problem because, as you can see, humans and fungi are evolutionary kind of similar, and there's a lot of shared pathways between them and a lot of these shared genes. But because it's a fungus, not human, there are of course genes that are specific only to the fungus itself. So we want to target these genes and also make sure that they are essential so that we can target them with these drugs and make sure that it's very effective. So with that, how do we even know what genes to really target with these antifungal drugs? Well, we have something called transposon sequencing. So as you can see, there's a red arrow, and that is basically a transposon insertion. And we have something called a transposon where it can be randomly inserted into that genome, and wherever it lands, it'll disrupt whatever is there. So if it lands into a non-essential gene, as you can see, neoformans still survives and is happy. But if it lands between genes, that's non-essential gene, it still survives and is also happy. And then with that, you have an essential gene where it landed, and because neoformans is normally a haploid and only has one of those gene copies, if you knock out and basically disrupt a essential gene that only has one copy of, it will die and it won't replicate. So we can basically map out all these different transposon insertions back to each other and then see where they all land, because if it can't replicate because it disrupted an essential gene, you can't really see that later on when you're sequencing it. So here we have a gene that is CNAG0597. And we actually identified this gene through transposon sequencing. And we saw that it basically had no insertions, which I will talk to you about later on. But what's interesting about this gene is that if you disrupt it, that actually may lead to more of that actual resistance to those antifungal drugs, specifically fluconazole. And here's some data on how we actually interpreted this gene. So as you can see, when I was talking about earlier, the gene, which is the green bar, there are no transposons within it, right? Nothing's landing there. And to even infer anything about the function of this, we can look outside of the actual gene itself, for example, in its regulation areas, like the 5' upstream region. And what's interesting is that compared to our normal growth medium, which is on top, that's the MSO, when we placed it in fluconazole, it actually tolerated more of those inserts in the regulatory region. So there's definitely a change in phenotype, and that is why it's interesting and we want to study it. So with that, we have established our target gene. Now, we want to go on to the main part of this project, something we want to look at, gene dosage sensitivity. So, like we said earlier, TNC, when you place it there and it has those transposons, it actually fully disrupts wherever it lands. And either fully disrupt an essential gene or a non-essential gene, and if it's a non-essential gene, you see a happy face and a high level of fitness throughout, even if it's disrupted. But if it's an essential gene, the fitness level, or basic survivability, drops all the way down. And because it only does full disruption, we only see the far right of that graph. But we also want to see what happens if there's a partial gene disruption, right? Because one of the important things that you need to realize about drug treatment is that it's never 100%. And that can be for different factors like the physiology of the specific person, for example. And we only see it through this lens through transposon sequencing all the way to the right. Basically absolute, but we can't see it in the middle. So wouldn't that be really cool to study? Wouldn't that be awesome and something that we should look at if we're trying to develop these antifungal drug treatments? And how can we do that? That also leads into the spectrum, like I said earlier, of essentially dosage sensitivity, where like on that graph, there are these different lines, even for all these essential genes, where it could be following a blue, red, or yellow curve, where, for example, the blue curve, if you have like even a mild disruption, fitness drops all the way down. But if you have this yellow curve and you start disrupting it, it will still maintain a high level of fitness as you keep disrupting it. So there can be an array of phenotypes, and we want to look into that. And how will we look into that? Well, we have basically these haploid neoformans, and that's how it normally exists in nature. But with these haploid neoformans, they could have an essentially same copy of that gene. So basically the exact same. And then when we breed them together and pause them in that reproduction stage, we can make a diploid, a homozygous diploid with the same exact gene copy, but twice, right? And then what's interesting is that this is basically a heterozygous diploid after we actually take away one of those gene copies. So it only has 50% of that gene dosage left. So basically 50% gene disruption. And with that, like the graph I showed you guys earlier, we want to see if there's a different type of phenotype, for example, like that blue line, where at 50% disruption, it may have something that's different from complete like drop in fitness or a complete like maintaining of fitness. So how do we even delete one of those gene copies? Well, we have this. For example, on the bottom, if you see that bar, that's called HYGR, that's a high resistance marker. And around it, those red regions right there, those are homology regions, where it's basically the exact same sequencing as the gene right there. And the gene will have the same homology. And so what we can do is that we can basically swap the gene with the resistance marker, and because we have the resistance marker, we can screen for that. And then after that, the resistance marker will basically take the place of that gene. And leading on from that, we were able to do that in that target gene we established earlier, which was again CNAG 05997. And then we want to now phenotype it to see that growth curve afterwards and that level of fitness, even with that disruption. So to do that phenotyping, we first have e-test phenotyping, where we basically incubate neoformans, then we dilute it, normalize it to a specific amount of cells / optical density, and then we spread that on growth media. And then afterwards, we add that fluconazole, which is the antifungal drug treatment that I said earlier, and then we see, as you can see right here, we want to record that circle. If you guys can see where I'm pointing, right there. And that's basically inhibition and areas of non-growth. And we want to see how that is compared to wild type, which is basically our normal, versus our actual mutants where we deleted that gene. So with that, here are some of the results. I believe you guys can be reading that right there, possibly. But as you can see, that circle across all of them is actually pretty similar, even though we did a 50% disruption and we deleted one of those gene copies. So that's interesting to see. And then, like I said earlier, we also want to continue to do more phenotyping. So we also have spot assays, where again, we are incubating the neoformans, diluting it, normalizing it to a specific amount of cells, and then adding it into a well, and then diluting it from left to right, so that we can see different amounts of cells on the actual antifungal growth media, where it's the same YPD, which is a rich media, where it has all the nutrients it needs, but also different concentrations of that fluconazole as well. And we want to see how well these different mutants grow compared to the actual wild type. So with that, here are the results for that again. As you can see, when I said YPD, that's our rich media, so it has all the nutrients it needs. But as we go from left to right, it actually has different concentrations of that antifungal drug. So we have 8 micrograms per milliliter, 13.5, and then 16. As you can see, it decreases in the amount of growth as you go further right. But these 4 bottom rows, those are all the independent mutants. And then the wild type is basically our homozygous diploid that we established earlier and made. So as you guys can see, it's actually pretty similar in phenotype. All of the 4 bottom rows compared to the wild type across all of them. So again, it's a similar phenotype. So in terms of interpretation for all those results, if you guys remember that graph I said earlier, where there was this yellow curve, even though we did a gene disruption, it actually maintains a high level of fitness comparable to the actual wild type, which is interesting to see, even though we classified it as a gene that is essential normally in the haploid. So that's interesting to note if we're trying to find antifungal drug treatments, because we want to find like these different genes where even if we disrupt it a little bit, the fitness level of neoformans will drop all the way down, instead of maintaining a high level fitness like this. So moving on, here are the future directions. So after we did that phenotyping, we still have to do some more steps. And the first step includes sequencing these heterozygotes of C90597, and then also doing a similar process in other genes like AFR1 and ERG11, and some other genes that are specific to fluconazole for comparison. Wait. There we go, wait. There we go, acknowledgements. Here, this is in the Billmeyer lab, so thankful for everyone that's in that lab. A lot of you guys are in the back right now. I can see you all right there. So I want to thank the PI, who is Dr. Blake Billmeyer, and of course, my mentor, Ms. Amy Kuhn right there. So, acknowledging all of that, thank you.